door, Doctor.
I'm sorry, Mr. White, but you've come down with a sudden case of diabetes.
Bacterial transformation, a genetic alteration of bacterial cells caused by an introduction of a foreign gene into bacterial plasmid DNA, can be used in the synthesis of pharmaceutical products.
How can we afford all these bills?
I think I have a method. I need someone's help.
Yo, Mr. White!
Don't I know you from somewhere?
Yo, Mr. White, doesn't matter. I heard you want to get in on that bacterial biz.
Alright, Mr. White, what do we need?
Alright, Jesse, here's what we need. First, we'll need a human insulin gene, micropipettes, E. coli bacteria, a petri dish for the cultures, restriction enzymes and DNA ligase, and finally, the ice and hot water baths.
Alright, so, Mr. White, once we have all this stuff, what do we do with it?
Alright, here are the steps.
First, we use restriction enzymes to isolate and splice the gene coding for human insulin out of a DNA sequence. Restriction enzymes are also used to remove a segment of the bacterial plasmid DNA, leaving room for the insulin gene to be inserted. As the insulin gene is inserted into the plasmid, it binds to the sticky ends of DNA, and is then glued in place by DNA ligase. This same process is used to introduce an ampicillin resistance gene into the bacteria. The bacteria is placed in a nutrient-rich culture and allowed to replicate. The antibiotic, ampicillin, is introduced to the bacterial colony. Any non-transformed bacteria are killed, while the transformed bacteria continues to replicate. Eventually, the insulin is ready to be harvested.
Calda estate non si riesce a far più niente che morire sotto il sole, tra le capole e la gente, e non so più dove andare, ma non voglio questa gente. Dove sei? Dove sei? Dove sei? Dove sei?
Alright, Jesse. Time to get cooking. This is our first batch. I need you to distribute these.
Hi, Mr. White.
Jesse, this is a great idea. We've made a million.
Stop right there, criminal scum! You violated FDA law on the product pipeline.
I don't know what the product pipeline is. What are its steps?
Oh, wow. A shame. Outstanding. Oh, I didn't see you there.
Step 1 of the biotech product pipeline is research and development. This process is very costly and time-consuming, taking 5 or more years and costing $155 to $225 million. Researchers, including biotechnologists and chemists, work to isolate and identify commercially viable products. Upon finding a potentially viable new compound, chemists work to mold the compound into a stable and safe form. Step 2 is the preclinical phase. Preclinical research may take up to 4 years. In this phase, researchers screen the potential drug with various different cell and tissue types to study its effects both long-term and short-term. These tests are conducted in both animals and human cells, so it can be determined whether or not to proceed with more advanced trials involving humans and animals. Step 3. Filing an investigational new drug application and completing clinical trials. In this step, a company must complete an IND application for the potential drug and send it off to the FDA and complete drug trials on living subjects. Clinical trials are divided into 3 phases, each containing a gradually larger sample of patients. Within these trials, researchers monitor patients exhaustively, identifying any side effects of the treatment. This step is the most costly, adding up to as much as $300 million. Step 4, filing a new drug application and scaling up production. This step finalizes the results of clinical trials. From here, the FDA determines whether or not to approve the drug. It can take as long as 6 years since there are so many products being pushed through the pipeline every year. Pending approval, the firm must develop techniques and procedures for large-scale manufacturing of the product. Once the drug pipeline is complete, the firm markets the drug to doctors and engages in advertisement.
The rules and
No,
I don't know what the
I don't even
pipeline
know
is.
what the product pipeline is. What are its steps?
Okay. Okay. Okay.